This is part 1 on a lecture on bacterial conjugation from April 8th, 2020. And the main purpose of this lecture is to define some terms associated with conjugation, such as sex pilus and F factor, and then talk a little bit about what F factor F plasmids are and what parts of these plasmids allow them to be transferred to new hosts through conjugation, as well as to integrate into the host cell's chromosome. And so most organisms generate their genetic diversity or genetic variation through the process of sexual reproduction. But as we talked about in the last class, bacteria reproduce asexually. And so they need another source from which to get genetic variation. And one of those sources is mutations. And the other comes from what's known as horizontal gene transfer, which is the transfer of a gene from one independent and mature organism to another mature organism. And what that means, in terms of the way we normally think about passing down genetic variation and genetic information, is from mother to daughter cell, right? Or in a vertical gene transfer from a mother to progeny. But with horizontal gene transfer, you would transfer genetic information from one adult organism to another, or from like mother cell to mother cell, for example. And there are 3 different types of horizontal gene transfer, which we're going to talk about in this class. But today we're going to focus specifically on the process of conjugation, or transfer of genetic information through direct contact. And so, as I just said, conjugation is a transfer of DNA or genetic information by direct cell-to-cell contact. And that cell-to-cell contact happens through a connection, as you can see here, known as a sex pilus. And in order for conjugation to proceed, the donor cell, or the cell that is going to be transferring its DNA to the recipient, must have what's called a conjugative plasmid. And to just kind of remind you what plasmids are, they're small double-stranded pieces of DNA. They're independent of the bacterial chromosome, and they can be inherited independently from mother to daughter cells, and they can also replicate independently. And so for conjugation to happen, or for DNA to be able to be transferred from donor to recipient, a donor has to have that conjugative plasmid. And the most well-studied conjugative plasmid is a plasmid from E. coli, known as F factor. And F factor contains many different genes. Specifically, it contains some genes that are needed for that cell attachment to form between donor and recipient cell. It also contains genes that are necessary for the plasmid, or the DNA itself, to be moved from donor to recipient. You can see a picture of F factor plasmid here, and then kind of a zoomed in view of a specific part of the F plasmid, which is known as the TRA operon, or the transfer operon. It contains a lot of genes. You can all see that they have this TRA nomenclature in red. And that's because they're involved in the transfer of DNA, right? And so some of these genes encode proteins that are involved in forming this pilus, or this contact point between donor and recipient cell. Some genes encode proteins that are part of the type 4 secretion system, which we'll talk about more in the next lecture. And then several other genes in this TRA operon encode enzymes that are involved in both bringing the DNA, that plasmid DNA, to the pilus, and then allowing it to pass through from donor to recipient cell. And in addition to the genes that are just involved in moving of the plasmid, making connections between cells, there are also some genes present on F factor that don't have those functions. But what they do function to do is to integrate that F factor plasmid into an existing bacterial chromosome. And so there are some types of plasmids, F factor is one of them, that can exist independently or alone, as well as within a bacterial chromosome. And those plasmids are what are known as episomes. An F factor is an episome, and it has a specific sequence known as the IS, or insertion sequence, that can line up with a corresponding insertion sequence in the bacterial chromosome and then result in integration of this circular effector plasmid right into that bacterial chromosome, as you can see on the bottom.